Hello everyone, nice to meet you. I hope next time everyone should be on time. It's time to begin our today's experiment. And first, I will conclude the result of the cytobiology of the last time. And this result just from one group of your experiment. I told you you can observe the color of all the cells and number the very darker cells in one group. And we can compare the glucose deprivation group to the control group. We can observe that there are less cells in the glucose deprivation, and the color of the cells in GD group is deeper than the control group. From this, maybe we can conclude that there are some Beck's conformational change in the glucose deprivation group. The same, we compare the DDP treated group to the control group. We can observe that the color increased, or we can see the stronger staining in the DDP treated group. Depending on the concentration of DDP, we can see with the increase of concentration of DDP, the color increased too. And from this, maybe we can conclude that during the process, there are conformational changes of Beck's. And from this, we also can observe the morphology changing of the cells. So compare the morphology and the color of the cells. From this, we can conclude that during the glucose deprivation and DDP-induced cell apoptosis, maybe can through the conformational changing of Beck's. However, the signal pathway is very complicated. So if we wanted to make a correct decision, we need a further experiment. And one of your students have explained this very well. He said we need to do some additional experiment. And here I want to mention one thing. That is, the color didn't represent the apoptosis. We can only make sure the cell underwent apoptosis through the morphology change. And another thing is some students wrote that because the cell apoptosis, so we can see the color increased. And that means because it apoptosis, so the Beck's conformational change. This is apoptosis, and this is the Beck's conformational change. And some students say because the apoptosis, so the Beck's conformational changing. However, from some picture we can know the conformational changing of Beck's is the upstream of activation of caspase. So based on this research, we think the Beck's conformational changing is the upstream of the apoptosis. This is one thing I want to make sure. Always some students asked me about their discussion is right or is wrong. I think the experiment of cytobiology is different from the math or physics. So you can observe your result and give a reasonable explanation. If you think this result is reasonable and you can get some data to support your explanation, it's okay. And so I think I just hope from the whole experiment of cell cytobiology, you can know if we want to make sure a phenomenon, we need to do a serious experiment to make sure. However, there are still something in your lab report. One student wrote just the object of last experiment is we used the SABC protocol to derive cell apoptosis and P53 presence. I don't know where the P53 came from because the objective of the experiment is just we want to make sure whether or not there are conformational change of bugs during the apoptosis. So I hope all of you more concentrate on the experiment. This is a result of our experiment of cytobiology, and we will go into today's experiment. The experiment of today is the chromosome binding, G binding. Can you still remember last week we prepared the metaphase chromosome? So today we will do something with the chromosome. However, we just harvest the cell from the culture cells. We use the HeLa cells. However, in clinical, most of the time we do not use the culture cell. Most of the time we will use the blood. So this is a procedure of venous blood. We get the venous blood and separate off the red cells because they are nuclei in the red cell. And we add culture medium to white cell suspension and incubate 3 days and 37 centigrade. And maybe during this procedure we need to add some factors to stimulate the white cell proliferation, and then it is the same as our last week's experiment. We use the cosamide or another agent, colchicine. This is similar, and then we separate off white cells, do the hypnotic silane added in the cells and fix the cell. Then cells spread onto slide by dropping, and last week we stopped here. And if we stand with the GIMSA, just use your slide with GIMSA, maybe we will get some result like this. Can you see? Then you can observe the chromosome spread if we stand our slides with GIMSA. However, we stopped here. What we want to do is just first I will introduce you something. We can see here, this is what we observed under the microscope. However, it's difficult to distinguish which chromosome is. So we just separated the chromosome and put them in line from the biggest one to the smallest one and grouped them. Several years ago, the scientists just use scissors, cut this, and then put this in line. However, now we just do the experiment with the machine. We can use some software to put the chromosome in line. So here we saw this inline chromosome, we give them a name, karyotype. So a pattern or picture of chromosomes from one cell grouped into pairs and organized by size is called karyotype. The normal human karyotype contains 22 pairs of autosomal and 1 pair of sex chromosomes. And so the normal karyotype for female contains 2 X chromosomes and denoted 46XX, and the male have both an X and a Y chromosome denoted 46XY. This is the karyotype. And we can see this picture under the microscope. We observed this, then we put the chromosome in line and grouped them. Then we can get the karyotype. This is a normal male karyotype, and this is a normal female karyotype. From this we can see we can distinguish some chromosome just like this, because this is the biggest from the size and the position of centromere. Maybe we can distinguish some chromosome. However, it's difficult to distinguish. We can see this group. It's difficult to distinguish one by one. So what should we do? Then scientists found, you can see here is the same chromosome. However, this chromosome we can saw there are some binding on some band on the chromosome. So we can distinguish the chromosome by chromosome binding. Chromosome binding is today's experiment. And each chromosome have a unique binding pattern. That means the first chromosome in the human being, in the A people or in the B people is the same band pattern. So we can distinguish the chromosome, and we can know which one is some abnormal, which one is normal. This is the chromosome binding, so this is a technique for staining chromosome so that bands appear in a unique pattern particular to the chromosome. And nearly all binding methods rely on harvesting chromosome in mitosis. That means the experiment of our last week is very important, because if we didn't harvest the chromosome, we cannot do today's experiment. And there are several binding methods. We will select one to do today. However, I will introduce several binding methods to you. First is the G-side binding. We also call it G binding. We can say this is the real chromosome, and this is the mimic one. The G-side binding is the most frequent used, because this sample can be keep for a long time. So the G-side binding is the most frequent used. And each chromosome pair stays in a distinct point of light, and you can see here dark and light bands. We call the dark bands G band, which are strongly AT rich. On the contrary, the light band we call R band, and which are strongly GC rich. This is the G binding. And another is Q binding. Q binding is a fluorescent pattern. and we use the clean for staining, and the band is the same of the G binding, because is positive one is the Q binding is the same of the G binding, the pattern of the bands. This is the Q binding. However, R binding is not the same. R binding is reversal towards G binding. That means the dark in the G binding and the positive in the Q binding. Here is a light or negative R band. And that means if in the GIMSA binding the band is light, then here it is the dark one. So this is R binding, also named because it is reverse binding, so we call it R binding. And another is C binding, centrometer staining. Because it is centrometer staining, we can see here the centrometer is dark. So from here we can clearly observe the centrometer, and this also stained with GIMSA. So we can use this to observe the centimeter, even the Y chromosome, because Y chromosome is also the dark stain. This is the C binding. And another, we just want to visualize the telomere. We can see the telomere, so this is called T binding, just a telomere binding. And the objective of today's experiment is G binding. We can see the picture here is normal human karyotype. However, this is different from the picture I just showed you, because that is just the chromosome, stained with GIMSA or other thing. However, there are no bands on the chromosome. However, you can see here is the dark and the light band on the chromosome, and here is the normal male, and this is the normal female G binding. This is the objective of today. We just want to the G binding, and the G binding can for the chromosome identification and karyotyping, and determine abnormality of chromosome number, translocation of material from one chromosome to another, and deletion, inversion, or application of chromosome segments. That means I just show you the normal pattern of GIMSA. One is a male and another is a female. We can compare the target chromosome pattern to this, and you will observe the different, and you will get some clue from this. And how to do the G binding? We just harvest the mitral phase chromosomes. So we treated the mitral phase chromosome with the trypsin, or particularly digest the protein and stained with the GIMSA. So this is the mitral what G binding we should do. This is a protocol. First, we just dissolve the 25 milligram trypsin in 50 milliliter 0.85% sodium chloride. Then this is our trypsin solution, and we put this. We will use this to make our trypsin solution, and that means we will put the 50 ml sodium chloride in here and add the trypsin here. Then we just put here in the water bath at 37 centigrade. And then we will add 2 drops of 0.4% phenolsulfoxylene. And the color will change. And we will adjust the pH to 6.4 to 6.6 with 5% sodium bicarboride. We will adjust this by the pH test strips. And then we will put the slides into the trypsin solution, just put here. And each group have 2 slides. You prepared 2 slides last week. So put into the trypsin solution 1 for 1 minute and 30 seconds, and another slides for 1 minute and 45 seconds. And because it sometimes the time will influence the binding, the result of binding. This is a protocol, and after this, we just rinse the slides in water. You just can use the sink in the rear of our classroom and place the slides in a coupling, and just you put the slides in the coupling like this. Just put there and pour the Gymstar working solution over the slides. Just cover the slides. After that, we steam for 10 minutes. After 10 minutes, we rinse slides in water again. After that, you just air dry the slides. That means do not so much water on the slides, just air dry the slides. Then we can check the slides using a microscope. Using the microscope is a little different because today we will use the oil immersion lens. So first you should use the low magnification lens. Then maybe use the low magnification lens. You can see just like some ants here, very small, just like this, under the microscope. If you saw this or another one, and maybe you can see some cells still there. And if you observed we call this chromosome spread. If you saw this, you just changing the lens into high magnification lens. And then you can observe. After that, you can change it into the oil immersion lens. That means this is our slide, just like this. And you observed one chromosome spread here, matrix spread here. Then you just drop one oil here. That means drop one oil. This is the oil. You just put the oil on your slides. And then you swing the lens here. Just make sure the lens is in contact with the oil. Can everyone understand here? Okay. Yeah, touch, contact, touch the oil. This is something I want to mention here. And then you can observe the result. After you observe, you should clean the lens with the saline. Here, you just clean the lens. And this is, maybe you can see here, this is a good result because you can see the clear, dark and light band on the chromosome. This is our result. And another question is what a karyotype tells us. We got the karyotype, then what we do? karyotype analysis determines the number of chromosomes in the cell and whether there are any pieces of chromosome material that are missing, extra, or rearranged. I will show you a picture. Can you see here? There is different from the picture I just showed you. That is the normal male or female G pattern, and this is different. We can see an extra 21st chromosome here, so this is a downstream here. So it's a different. Today you can number the chromosome. You can count how much numbers the chromosome in your experiment. And there are something to mention is the trypsinized time. If the time is long, that means over-trypsinized. The over-trypsinized chromosome appear fuzzy. Then it's difficult to recognize the exact bands. And if the time is shorter, that means under-trypsinized chromosome, and will have indistinct bands. Deserves contract, very difficult or impossible to determine the bands. That means long or short time will influence the band. So the adequately trypsined chromosome will show telomeres not overly digested. So you can observe your chromosomes, and the G bands will appear sharp and in contrast. Okay. This is all the experiment.